This conference will now be recorded.
I'm just always ready to go if I forget to hit the record button. So thank you, Tamara, for reminding me to record. Okay, so I know some of you may be on spring break right now, doing fun stuff, and you may be tuning in. Hopefully you're not. Hopefully you're enjoying your time off if you're on spring break with your family and your loved ones. But for those of us who are not, who are still out here in the streets, hustling, trying our best to do what we need to do. Welcome to another exciting day. So today we have to talk a lot about microbiology, so it's going to be exciting, but we do need to cover some just some general things. All right, so we're going to do some infection prevention updates, your study plan really quickly, and we're covering parts of microbiology basics and laboratory diagnosing. So that should be chapter 26, not 22 on there. All right, again, Google Drive. Please make sure that if you do not have access to the Google Drive, that you get access to the Google Drive. The link is right there. You're able to access so many of our materials on there, all of the things that you need. I can post the link at the end, just so that we don't get distracted. But there's so many different resources in here. I actually don't even know everything that's in here. I can only speak to what's in the Florida materials folder, but that's where you can find our materials. All right, YouTube resources. People are still asking me lots of questions about where can we find the recordings for our meetings. I cannot send out emails every week. I'm so sorry, it's just not feasible. There's a lot that's going on. A lot of projects at work, and so they will be uploaded to YouTube. You can go on YouTube and watch the ones that we've done so far. That for the past 2 weeks, you're able to watch them on there. Okay. This is Kearns Rivers, the Kearns Rivers CIC study group, which is great. So if you are interested, lots of good videos on there. And then the CIC epidemiologist is where I'm posting our meetings. Okay, another resource for you guys. How many total sessions will we have together? That's a great question, Dorian, because I really don't know. So I have a plan, but here's the problem. I'm already messing up my own plan, because I thought, okay, let's just head straight into week 3 tomorrow. But I really think we need to cover more of laboratory diagnostics and antimicrobial stewardship. So next week is actually going to be 2.5. And we're going to have to reschedule from Friday to probably Thursday. I will have to send out an email about that. But I have a huge nursing leadership meeting that I need to attend on Friday. So I will not be able to meet next Friday. But anyways, you guys will get an email. Don't fret, don't worry. If you can't attend, it will also be recorded. So don't stress, okay? It's going to be great. It's going to be fine. So Seattle, Washington, Patty Montgomery. She's amazing. She is amazing. This is another study group that you can join that is question based. So all she does is just questions. It's a great way to test your knowledge, to have some fun. You are able to register for her session. I will also drop the link. I need to make a list of all the links I need to drop because, okay, so you need Google Drive. And then Patty. Okay, excellent. You know what? I might just send out an email because this list is already getting long. But as you can see, Wednesdays from 12 to 1, no cost, no judgment, no preparation. You can just register, go ahead, sign up, test your knowledge. She's great. Okay, for those of you who may not be aware, the SHEA Spring Conference is happening in 24 days. It's going to be in Colorado. How many of you are attending? Are any of you going? Society for Healthcare Epidemiologists of America, SHEA. Anyone going to SHEA? Okay, I'm not getting any answers. I'm not going to SHEA, but I just wanted to let you guys know it's all because I'll be attending APIC in the summertime. But I wanted to let you know that there is a conference, especially for those of you who may be in Colorado or near Colorado. It's a fantastic conference. I loved attending SHEA. I had a really great time when I went a couple years ago, and it was absolutely amazing. Yes, okay, so Dorian, I'll be at APIC as well. Rebecca's going to be at APIC. Excellent. I'm going to be able to maybe see some of you this summer. That's great. SHEA also has a prevention course in healthcare associated infections. It's $25. Just wanted to let you know that there are resources for those of you who may be studying or who need additional, you know, knowledge, classes, interactive ways to learn. This is another option that you have. They cover lots of great things: prevention of CLABSIs, CAUTIs, SSIs, VAEs, C. diff, MRSA, all of that good stuff, disinfection and environmental control as well. Okay, next thing. How many of you guys have heard? Let me mute. Okay, how many of you guys have heard about CDC's Project First Line? You can just in the chat, yes, 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 no. If this is the first time that you're seeing it, okay. Okay, if this is the first time that you're seeing it, just feel free to dive right on in. So I'm going to read you their introduction. So it was created with healthcare workers, for healthcare workers. Project First Line's resources are responsive to the unique needs and time constraints that result from working in a healthcare setting, ranging from bite-sized social media graphics to toolkits for healthcare workers interested in hosting their own infection control trainings. The variety of formats helps ensure all healthcare workers can easily access the infection control information they deserve in a way that works best for them. So when you type in CDC Project First Line, they're going to have a lot of different resources. I had a lot of fun going through the resources. One of my favorite images was this one because it's just fantastic. I don't know who did it, but they deserve a prize. There's more to water than meets the eye. Germs can grow in water and wet places like sinks. Those germs can spread to other surfaces, devices, and people. And so you guys know how I feel about water-loving bugs and sinks and splash zones, plumbing, all of biofilms. It just gets me riled up, okay? It just gets me all sorts of, it's just so much fun to learn about it. The papers that have been published on it, it's amazing. But anyways, back to Project First Line. So they have all of these different posters and resources. Don't have audio on my end. Unsure if it's just me. Can you guys hear me? Okay,
Can.
so
Yes.
so getting lots of yeses. Okay, so it might just be you. It might just be you who is not getting. Oh well, that won't be helpful because I'm talking if they can't hear me. All right, you know what? We just gotta keep on.
Those are you recording or not yet?
Yeah, I am recording.
Thank you so much.
You're welcome. So posters. You've got posters. Let me message this person. Others can hear. Okay. All right. Next. So germs live in water. Germs live on skin. Lots of different resources for us IPs as when we're getting ready to put presentations or do education. We're always looking for different images or access to educational material. They have entire PowerPoint decks that they've already put together that talk about all different sorts of stuff. I mean, there were two interactive, one that was like about diarrhea and the other one that was about, I believe, vascular access, which I didn't get to do, but I'm gonna go in and explore. So I'll let you guys know, but you should explore as well. Next thing, we've got our NHSN training next week. Is everyone signed up for my IPs, my infection preventionists, our 2022 virtual training? That's next week, Tuesday, March 22nd through March 24th. If you guys are not aware, your system may be like throwing it to spam because it's coming through vFairs, which is different than what NHSN usually uses, but it's legit. It's legitimate. They sent out an email saying like, hey, this isn't spam. We're not phishing. Like, this is the platform that we're using to do our training. So please make sure that you attend. So they're going to be going over lots of different stuff. The first day, so March 22nd, it's going to be a lot on CLABSIs, BSI exclusions, case studies, secondary BSI, and NHSN site-specific infections, which CLABSIs for me are, so the two most time-consuming infections to work up personally, personal opinion here, are CLABSIs and surgical site infections. And so, but CLABSIs are so much fun. Like, that's when you really get to be Inspector Gadget. That's when you really get to get into that chart, try to find your secondaries. It is an investigation situation. It is so much fun. They have a registering link. Just type in CDC NHSN 2022 training. There's a registration link so that you can learn all about how we make our determination and exclusion criteria. Okay, enough of that. Moving on. So practice exam 1. If you have not taken your practice exam 1, please make sure that you get that done as soon as possible, because your practice exam 1 should be guiding your studying. Okay, this is an example of somebody who completed their practice 1 assessment, and then you can tell, okay, the percent correct. What areas did they score really low in? Well, one of the areas this person scored really low in is identification of infectious disease processes, which lets that person know, hey, I need to put more effort into this specific portion of the test because it's a big chunk of the exam, and I need to make sure that I have an understanding of where I'm lacking, what I need to improve on, what additional chapters I need to read. All right, why is taking a practice exam in the beginning important? It provides you a baseline of your current strengths and weaknesses, helps to guide your exam preparation, focus your energy on concepts that you continuously miss during the exam, select chapters to read that you commonly missed. Timing. Timing is so important for the exam. If you don't time yourself properly, you're going to run out of time. I can't tell you how many people send me emails saying, I ran out of time. I had to guess on the last 15 questions of my test because I completely ran out of time. I spent too much time either overanalyzing the questions. You didn't do your test prep appropriately, where it included taking practice exams, timed practice exams in really quiet locations. All of this is really important to prepare for the exam well. Mental stamina. Let me tell you something. Certification exams will just really run you down mentally. You have to, it's just like constant thinking. You're just constantly working through all of the files in your brain, all of the knowledge that you have to pull out that piece of information that you need for this test. And so it helps you build your mental stamina and familiarity with question structure. All right, so today we're going over chapters 24, Microbiology Basics, chapter 25, Laboratory Testing and Diagnosis. And we will not be covering chapter 26, Antimicrobial Resistance. We'll go over that next week. All right, Microbiology Basics. So microbiology easily has to be one of my favorite classes that I've ever taken. It is so much fun to get into a lab and to play with bacteria and Tryptococcus agar, growing all sorts of different stuff, learning about tests, and actually practicing what you're learning about. So actually doing the gram staining and just getting the opportunity to see what people who actually work in the lab and do this day in and day out do. So it's one of my favorite classes. And it's very important. Microbiology is very important for the infection preventionists. You have to be very familiar with lots of different infectious diseases to make sure that you do your job well. So it's fun. You need to have fundamental knowledge of microorganisms, their identification, significance, and basic laboratory techniques they provide the infection preventionists with an understanding of pathogenic microorganisms. Basic laboratory techniques. Are we going to get into all of the details of everything that can happen within a microbiology laboratory? Absolutely not, because we would get lost in the sauce. All right, we would get lost in the sauce. Let me tell you, there's this man, his name is Dr. Alexander, and he is so smart. He's the director of the microbiology lab, and the amount of knowledge that this man has is just ridiculous. He can tell you the difference between like a MALDI-TOF and all sorts of other different things that personally, I just ask him. If there's something that I don't know, I just ask him or other people. And that's important too for IP. You don't have to know everything. It is not your job to know every single possible thing within the spectrum of our role. You have to know who your resources are and who you can go to for questions. Whether it's your director of the lab or your antimicrobial stewardship pharmacist, you have to know who your resources are. The clinical microbiology laboratory can provide information on microbes determined to be of clinical significance. Proper specimen collection and transport are integral to the recovery of valid microbiological information. This is very important. Specimen collection and transport will be on your test. I'm going to say that once and then I'm going to say it twice for the people in the back. Specimen collection and transport will be on your CIC exam. You need to know it. We were going to cover it today, but you need to know it. Also, if you're coming on the meetings and you're learning and you're having a great time, that's wonderful. Remember, our meetings are supplemental. You still need to be reading your assigned chapter readings to make sure that you're actually getting all of the information. I cannot tell you all of the information that's in that text because we don't have enough time. Okay, essential knowledge for IPs. Bacteria are astoundingly small. They're invisible to the naked eye. You know, we can't tell that that mouse that we're about to touch has Staph aureus on it. We just can't tell. They're invisible to the naked eye. So they're very, very small, and that's important for us to remember. Some bacteria can transform into endospores. Okay, so here's different examples of some endospores. So Bacillus anthracis, which causes okay, Joseph or Benson out here just going through the whole list, didn't even give the rest of the group some time, but good job. He was like anthrax, tetanus, pseudomembranous colitis, gas gangrene, botulism. Good job. Yes, so Bacillus anthracis, anthrax, Clostridium tetani, tetanus, Clostridioides difficile, obviously can cause pseudomembranous colitis, but C. diff. Clostridium perfringens, gas gangrene, and then Clostridium botulinum, which is botulism. Microbiology includes the study of bacteria, fungi, mold, yeast, protozoa, viruses, and algae. Okay, there is so much that you need to know for your day-to-day work when it comes to pathogens, because as IPs, we are reviewing labs constantly. I mean, we're having faxes come through our surveillance systems in the computer, whether you're using Cerner or Epic or whatever other electronic medical record system you're using. And so we're the people who are reviewing all of those labs, making sure people are properly isolated, that there's nothing that we are missing or that we need to be protecting our staff or patients from. So Clostridioides difficile, I wanted to talk about this one specifically since it was mentioned as one of the spores. This is the antibiotic resistance threats document that was published by the CDC back in 2019. If you're not familiar with this document, I really recommend that you take some time to read through it. It's about 150 pages, just a bit of light reading. In the words of Hermione Granger. Okay, just a little bit of light reading, but it's great. It's got lots of images. It contains a lot of really important information, but it is an urgent threat level as far as antibiotic resistance in the United States. And it can cause life-threatening diarrhea. Infections occur most often in people who have taken antibiotics for other conditions, and it is the most common healthcare-associated infection. So it's important to know about our spores. With that being said, here's our endospore formation. This is such a wonderful slide because I feel like we sometimes, we're just talking about spores, but then we don't really think about the struggle, right, that some of these friends, these bacteria, have to go through. I mean, yeah, it's rough out there for them. But here you have an example of what an endospore looks like. So you've got the vegetative cell that begins to be depleted of nutrients. So something in the environment is no longer serving that friend, that bacteria, and so its nutrients are starting to be depleted, and they're like, oh hey, okay, we need to do something about this. Which is when your chromosome is duplicated and separated. The cell is separated into a sporangium and a phorospore. The sporangium engulfs the phorospore for further development, and then the sporangium begins to actively synthesize spore layers around the phorospore. Then you follow this same trajectory all the way through till you end up with the core of your spore and your endospore, which is that little bit, that part of the cell that's trying to, you know, use the nutrients that are left to go into essentially a hibernative state, so that when conditions arise and they are great again, it's able to multiply and do what it needs to do. Which is why C. diff can live in the environment for about how long? Five months. Very good. All right, next we need to talk about our bacterial shapes and arrangements. Bacterial shapes and arrangements are a lot of fun. They're also very important because when you're reviewing microbiology results, guess what? We have a lot of preliminary results. And what do those preliminary results often tell you? Basics, shapes, stains, gram-negative rods, gram-positive cocci, gram whatever. They're describing to you what is already growing, because we haven't been able to speciate whatever it is that's growing in this culture. But hey, here you go, you get the basics. You know that there's gram-positive cocci growing in this. So immediately, as an infection preventionist, you should be thinking, okay, what different pathogens am I thinking about that could be gram-positive cocci, right? Or what pathogens am I thinking that could be gram-negative rods? So this is your bacterial shapes and arrangements are very important for you to know. They also help pharmacies start guiding what are we doing, where are we be going therapy-wise, okay? So for A, you have coccus, which is that spherical rod or bacillus. You've got your Vibrio, very commonly Vibrio cholerae, which is cholera. Spirillum. Then you have got your spirochetes. So can someone tell me an STD that's a spirochete?
Syphilis?
Treponema pallidum. Syphilis. Very good. And then you've got your branching filaments. So you've got lots of different options here. I wanted to include Neisseria gonorrhea because if I'm going to bring up syphilis, I might as well bring up another one of our urgent threats. So Neisseria gonorrhoeae causes gonorrhea, a sexually transmitted disease that can result in life-threatening ectopic pregnancy and infertility, and can increase the risk of getting and giving HIV. So important, we got to know our antibiotic resistance threats. So here we have our bacterial arrangement and size. So monococcus meaning 1, diplo 2, diplococcus, tetracoccus. You've got your bacillus, your diplo bacillus. So with those descriptions, you're having your singles, your pairs, which are diplo, your chains, which are strepto, your clusters, which are staph, your palisades, which don't necessarily have you know, like a set structural arrangement. So you've got lots of different shapes that you need to be familiar with, so that when the lab is describing what's growing on a plate, you're able to already start thinking of, okay, what am I really looking for here? What is it that I'm seeing, and what can I expect for this patient to grow? Right? Oh, gram stains. Gram stains are so important. This is going to be one of the first ways that you're going to be able to differentiate bacteria from one another, which is going to be that designation of either gram-positive or gram-negative. Okay, so there's a major difference in between a gram-positive bacteria and a gram-negative. What can you just visually look at and be like, yep, okay, this is very different? On the structure of that cell wall. What are we working with here? All right,
Color.
So we have color. Okay, I'm getting lots of other messages in here. So we've got permeability, peptidoglycan layer, thickness of the cell wall layers, lots of really good points that everyone's bringing up. But yes, peptidoglycan, your peptidoglycan layer in your gram-positive organisms is significantly larger than the peptidoglycan layer within your gram-negative organisms. You also have different components of those cell walls. So within your gram-positive cell wall, you've got your lipoteichoic acids. Within your gram-negative wall, you've got your lipopolysaccharides, your LPS, your lipopolysaccharides, which are really important because those are going to contain your O antigens. They're going to contain your lipid A, just so, which is a really important component for immunologic response, because when our macrophages start to break down gram-negative bacteria and that lipid A goes into the bloodstream, it can set off all different sorts of issues, fever, leading up to septic shock, right? So important to know pieces of, okay, you don't have to be a microbiologist, right? Do we have IPs who are microbiologists? 100%. Am I a microbiologist? No, I am not. Julia is a microbiologist. Excellent. So, you know, Julia could probably get in here and tell us all the details, all the tea on the gram stains, which 10 out of 10. Yes, because we love diversity in the field of infection prevention. We have microbiologists are in the house, okay? They are literally, Nahid's also a microbiologist. We've got really intelligent, beautiful laboratorian people here on the call today. So what I'm trying to say is you need to make sure that you know some of these basic concepts related to the differences between our gram-positive and gram-negative cells because, sorry, bacteria, because it's going to be on the test. They're going to ask you questions about the difference between gram-positive and gram-negative because it's one of our biggest differentiating factors between bacteria. And it's one of the very first things that you're going to get in a preliminary lab result, which should already start to guide you on, okay, what am I thinking here, right? Not just you as the IP, but also the clinical team, your doctors, your pharmacists are already going to be starting to think of, okay, I've got gram-negative rods. This is what I'm thinking about. Because all of these little pieces that we're discussing here, the lipoteichoic acid, the lipopolysaccharides, affect our mechanisms of how antibiotics act on these different types of bacteria. Now, again, I am not a pharmacist, so can I tell you every mechanism of action? Never, never could I. It's just not going to happen. But I know people who can. Okay, so it's all about knowing your resources and making sure that you understand the basics for your tests. Okay, so what do I have here? An important polysaccharide present in the gram-positive cell wall is teichoic acid. It acts as an antigenic determinant, so it is important for serologic identification of many gram-positive species. Okay, that was a lot. I get very passionate about gram stains. Speaking of which, gram stains, shout out to Hans Christian Graham, because we would not have the gram stain without you. So thank you for what you've done for the people. So it was developed in 1984. It delineates our two major groups of bacteria, gram-positive, gram-negative, and the differences lie in the structure of the cell wall. So again, our peptidoglycan layer. And here we have the chemical reaction of what's happening in the cell wall. So when you do a gram stain, you're using two dyes, which is going to be your crystal violet and your safranin. So what this little graphic here is showing you, if you've never done this, right? If you've never taken a microbiology class, you haven't had the pleasure of playing with bacteria and slide sets and crystal violet and safranin and agar plates, that's okay. Go to the lab, right? You have a lab, or you have a friend who has a lab. If you're in public health, you've got public health labs. If you're in the hospital, you've got labs that you're sending your tests to. Go spend, you don't have to spend the whole day, just go spend 4 hours learning from what they're doing, actually see them do a gram stain. There's nothing like being immersed in that environment and getting the opportunity to do things or to observe things, to shadow, to learn. We have so many resources working in hospitals to further our knowledge and to perfect our craft of being infection preventionists. Take advantage of every single opportunity that you can. Every single opportunity that you can. You can always learn, you can always grow. You should always be learning and growing in this profession. It's my favorite thing of being an IP. I have the best job in the world. Biased, obviously, but that's how I feel. Okay, so back to this. So with your gram stain, you've got your crystal violet, which, all right, so you spread your stuff. Is it pus? Is it sputum? Is it a vaginal secretion? It could be whatever you need it to be. So you put it on there. All right, then you pour your crystal violet over it. Both cells affixed to the dye. Then you run it water, iodine. That dye complex is trapped in the wall. For our gram positives, for the gram negatives, there's no effect of the iodine. With the alcohol, the crystals remain in the cell wall, doesn't come off, and the outer membrane for the gram negative is weakened and the wall loses the dye. So boom, no dye for our gram negatives. Then you take your safranin, which is your last stain, and you use the red dye, which is masked by the violet because of that thick peptidoglycan layer. And then the red dye stains the colorless cell for the gram negative. So also, you don't have a lab. You don't have a friend with a lab. YouTube. I don't want no excuses. There are so many resources to learn, to watch. You can see a gram stain done on YouTube, and I'm telling you, seeing things will put what you read into perspective. All right, so let's do a quick concept check. In a gram stain procedure, gram-positive bacteria stain purple because they have lipopolysaccharide layer in their cell wall that is decolorized with alcohol. B, their cell walls contain long-chain fatty acids that take up crystal violet easily. C, they have a thick peptidoglycan cell wall that retains the primary stain during the alcohol decolorization. Or D, gram staining is simple staining, so the only stain used is crystal violet. All right, so let's see what kind of answers I'm gonna get. This is what's gonna tell me whether I did a good job explaining gram stains or not. Okay, let's see. Okay, yes, very good. Very good. Everyone has put down C. Okay, this is great. This is excellent. So, yes, correct. They have a thick peptidoglycan cell wall that retains the primary stain during the alcohol decolorization. So, they have a lipopolysaccharide layer in their cell wall that is decolorized with alcohol. What is this referring to? Not gram positive, gram negative, right? LPS, lipopolysaccharides, they're going to be in our gram negative organisms. So that immediately, you don't even got to finish reading that sentence. You can just be like, nope, wrong. Their cell walls contain long chains of fatty acids that take up crystal violet easily. Long chains of fatty acids, what are we referring to when we're describing a cell wall with long chains of fatty acids? I can tell you that it's not gram-positive bacteria. It's what? Oop. Joseph again. Fastidious bacteria, acid-fast bacteria. Very good. And then gram staining is a simple staining, so the only stain used is crystal violet. That's incorrect. We use crystal violet and safranin. All right, good job. How do I go next? Do I have to turn off my pen? Oh, this is too difficult. Oh my goodness. Normal non-drawing mode. Okay, there we go. I figured it out. Everyone just hold your horses here. Oh, what happened? Erase all drawings. There we go. Okay. Technical difficulty, but I figured it out. Okay, another concept check. You have isolated a bacterium from your skin. Chemical analysis shows that it contains proteins, peptidoglycan, lipids, DNA, and teichoic acid. What sort of bacteria is this? Is it gram positive, gram negative, or I cannot tell from this information? Okay. Very good. Lots of you are telling me A. Yes, absolutely. this is our gram-positive, this is our image of our gram-positive cell wall. We've got that thick peptidoglycan layer. We've got our teichoic acid, which we know is really important when it comes to our gram-positive bacteria. So, yeah, good job. Okay, next thing that we're going to talk about is going to be our acid-fast bacteria or mycobacteria. Again, we're talking about those long chains of fatty acids. They contain mycolic acid, which is the cord factor. It's a waxy surface impervious to chemicals or dyes. So, you know, our mycobacteria are, we got to use a different stain. They're, okay, so when you read some of the microbiology texts, they'll tell you that they weekly stain gram-positive, weekly, like very weekly, which I don't know exactly what that means because I never did a gram stain on mycobacteria in my class. We only did, we only looked at acid-fast staining. So can't really, you know, can't really say too much on that. But yes, important pathogens that are in the that are acid-fast bacteria: tuberculosis, leprosy, opportunistic wound infections, and they do grow inside of macrophages. Mycobacteria are a lot of fun, but you do have to know your differences. And this is something that I've gotten to learn a lot more about being an IP and getting to collaborate with infectious disease physicians and really talk to them about their thought process when they're trying to rule out, you know, when we have a sputum that comes up and it's acid-fast positive, or when we have a, you know, they drained fluid from, literally, it could be anywhere. They drain fluid from somewhere and they're talking to me and they're like, oh, well, you know, I'm actually thinking that it could be this, 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 and this because it's already growing, so it must be a rapid grower. So this is what I'm considering, and it's really interesting to be able to see all of their thought process. And I've gotten to gain more experience with mycobacteria being an infection preventionist. So it's important to know our mycobacteria. So here is our acid-fast stain. So again, these contain long chains of fatty acids that make them impervious to crystal violet and other basic dyes. So heat or detergents can be used to force dye into this type of cell. And these mycobacteria, or the acid-fast stains, are very useful in identifying Mycobacterium species as well as Nocardia and Actinomyces. So here we go. You apply your primary stain of carbofusion for about 30 seconds. So here we go. We're applying our carbofusion. Then you're going to heat fixed cells to the slide using a flame. So you're using that heat to force that dye in there. You're going to decolorize with acid alcohol for about 15 to 20 seconds. Boom, boom, boom. We're decolorizing, and then now all of a sudden we're starting to see them. And then you apply your counter stain of methylene blue for 30 seconds, and then rinse excess stain. And there you can see your acid-fast organisms and your non-acid-fast organisms. So again, if you work in a lab, great, excellent. You can get the opportunity to see this, but if you don't, go out, find a way to make this information stick. All right, time for a challenge. Okay, so you've got your different types of mycobacteria at the top. And then we've got our different descriptions of that mycobacteria at the bottom. Let me check the time. Okay. So number 1, it's skin and soft tissue infections associated with fish tanks. Associated with fish tanks. Which one are we going to go with? Which one are we going to go with? Okay, excellent. Excellent. Good job. Mycobacterium marinum. Very good. All right, this next one. Rapid grower and often a water contaminant. It is part of a group of environmental mycobacteria and is found in water, soil, and dust. It has been known to contaminate medications and products, including medical devices. It causes chronic lung disease, disseminated cutaneous disease in immunocompromised patients, and post-traumatic wound infections. So which mycobacterium are we talking about there? It's not leprae. It's not leprae. So you can cross that one out. Which one do you think it is, Tamako? Obsessive? Good job. Good job. Good job. Mycobacterium abscessus. Very good. All right, next. A rapid grower is often found in water and sewage and will cause occasional opportunistic infections. Good job, Julia. Do you want to come off speaker and tell off mute and tell me how to say this one? Because I'm not sure how to say it.
I think that it's Kilone.
Oh, thank goodness, because I was about to butcher that one. Chelonier. Okay. Anyways, we have a microbiologist on the line, guys, so you heard it here first. Okay. Very good. All right. Next one, number 4. A slow grower. A slow grower. That is a water contaminant and may cause disease in patients with severely impaired cellular immunity. A slow grower. Okay. Very good. So Mycobacterium, that's another one that I'm not sure how to say appropriately. So Julia, if you want to do the honors.
Kansas SCI Immunology.
Oh, there we go. So a slow grower that is a water contaminant may cause disease in patients with severely impaired cellular immunity. And then our very last one, Hansen's disease. It can affect the nerves, skin, eyes, and lining of the nose or nasal mucosa. And it may take up to 20 years to develop signs of infection, so Mycobacterium leprae. All right, next one. Okay, we've got our mycobacteria again. So for this one, we're gonna go in order. So we're gonna start with Mycobacterium abscessus. Is Mycobacterium abscessus a rapid grower or a slow grower? Go ahead and put your answers in the chat. Mycobacterium abscessus. All right, rapid grower. Okay, next one, Mycobacterium. I already forgot how Julia said this one, but the orange one.
Kilone.
We're gonna have to practice this one, guys. I'm gonna make this the assignment for the next week. Sorry, my team's here, and so we're all learning. But she didn't say Shalone. She said Cologne. No, I don't think it. There's not a sh sound. Okay. All right. Yeah, the orange one. Good, good, good. Okay, next one. Mycobacterium. Can inside that one. It's kind of like Kansas with the eyes at the end. Joseph said slow. Everything goes slower in Kansas. Ooh, okay. Okay. Listen. Joseph's driving 16 to 30. I'm gonna need him to slow down today. Okay, next one. Mycobacterium chimera. Is it rapid or a slow grower? Rapid or slow? All right, we've got somebody that's saying slow. Yes, go Eva. All right, next one, Mycobacterium fortuitum. Mycobacterium fortuitum. It just sounds happy. Rapid, yes. Mycobacterium avium complex. MAC. MAC. Is MAC slow or is MAC fast? Slow grower. Very good. And then the last one, Mycobacterium mucogenicum. Which one do we think that one's going to be? All right, Eva said rapid. Eva, are you in the lab as well? Are you a microbiologist? No.
No, I'm a novice IP, but I'm studying.
Oh, good job. Good job. Okay. All right, Crockaflor white stain. What are we looking at here, guys? when we're using a calcofluor white stain, what are we looking at? I'll give you a clue. The first picture has a name right under it. That should really let you know what we're looking at. Okay, we're getting lots of different answers. Fungi, yeast. Good job. Calcofluor white is a special fluorescent stain that binds to the chitin in the cell walls of fungi. Adding calcofluor white stain to the slide will cause the fungi to become fluorescent, making them easier to identify under a fluorescent microscope. All right, next we're going to talk about bacteria and genetic diversity. We're going to have to pick up the pace here a little bit because I want to make sure we do some questions. So the genome can occur not only by mutation, but also by transfer of genes from other bacteria. Acquisition of an R plasmid can render a bacterium and its descendants immediately resistant to antibiotics if resistance genes are encoded on the plasmids. And then plasmids are DNA molecules that are generally circular and not essential to the microbe under some environmental conditions. So over here, you've got an example of conjugation, how a resistant gene moves between bacteria. The cells come in contact, a process called conjugation, and the plasmids move from one to another, taking the resistance gene with them and making the new bacterial cell drug resistant as well. And this is really important. I had a really wonderful experience it's like my first love in public health, which was my outbreak of Vim producing Pseudomonas aeruginosa, which I worked with my colleague Danielle Rankin, who by the way is completing her PhD at Vanderbilt. Everyone send her lots of good energy. She should be done this summer, and then she's going to be Dr. Rankin, which I'm not going to talk about too much because then I get emotional because I'm just so proud of her. But anyways, I really got the opportunity to learn about different mechanisms of resistance during that outbreak, and it was really interesting to see how, specifically in this outbreak, we were following a paper that was published in France with the use of bleach, pouring bleach, like closing the P-trap, pouring bleach down into the pipes to see if that could help with removing some of the biofilm, killing some of the bacteria that was in that part of the plumbing. What we actually ended up finding out was that when we did this, it made the bacteria in there very angry, and they started exchanging information so that when we took cultures of the plumbing again, we had serratias that were now having KPC. It was Hamas expressed in there. And so it's really interesting when you really get into more of this bacteria genetic diversity and where we're heading. I know that there was recently a paper that was published talking about some of the genetic diversity in the plumbing in India, but we don't need to. Anyway, you guys need to keep me on track because I get really fired up about biofilms. So back to this: transformation, conjugation, and transduction. Transformation occurs when naked DNA in the environment, possible from dead bacteria, enters another bacterium, so you're inheriting. Conjugation occurs when all or part of a plasmid is transferred from a donor to a recipient cell. The cell must be in direct contact, and transfer occurs via the sex pilus. Conjugation can occur between widely separated species, leading to rapid dissemination of genetic information. Scary. Transduction occurs when bacterial DNA is transferred from a donor cell to a recipient cell via a virus capable of infecting bacteria. Do I think they're going to sit down and ask you the difference between transformation, conjugation, and transduction? No, not necessarily. But it's important for you to know what is contributing to the antibiotic resistance that we're experiencing in our healthcare facilities. Antibiotic stewardship is one of the pillars of infection prevention, and so having an understanding of how this is happening and these different the genetic diversity or the different things that can happen for bacteria to gain antimicrobial resistance is important. Next, viruses. Viruses are neither eukaryotes or prokaryotes. They are obligate intracellular parasites, meaning they need to reproduce inside of a host cell. Okay, they're obligate intracellular parasites, which means they reproduce within living cells and are depending on the cell's synthetic and metabolic machinery. So, public health virology in grad school, great class, fantastic. I remember we got into a really intense conversation about whether viruses are alive or they're not alive, or, you know, et cetera, like how does it work? Are they in the in-between place? Is it a gray zone? Important for you to know, they just can't replicate on their own. What type of microscope do you utilize to look at viruses? They're so small, so you just can't use a regular microscope. You have to use I'm looking for a very specific word, very great, Joseph and Julia, electron microscope. Excellent. Virion, it's the outside the host cell. It's metabolically inert and does not grow or multiply until it enters a host cell. So that's the virion. There are 3 major methods to diagnose viral infections. 1, direct detection in the clinical specimen. 2, specific antibody assay to detect viral antibodies in the serum. And 3, viral culture. Viral culture, which is the most difficult. It's hard to make these puppies grow. So, diagnosis of viral infections. You've got electron microscopy, your ELISAs, which are your enzyme-linked immunoabsorbent assays, latex agglutination, DNA probes, PCR for DNA detection of viruses. Think about our respiratory panels, right? Our respiratory PCR that has all of our lovely different viruses, rhinovirus, enterovirus, parainfluenza, metapneumovirus. It keeps going. The list goes on and on. Adenovirus. Yes, PCR. So OIA, or optical immunoassay, which is antigen-based test that produces a reflection change for detection of influenza viruses. Light microscopy cell scrapings from infected sites that can detect cowdery type A inclusion bodies from herpes simplex virus. Pap smears, Papa Nicolau. That's how my mom always says it in Spanish. Number 9, Negri bodies for the diagnosis of what? Yes, correct. Rabies. And there we have an image from Wikipedia. All right, microscopic slide preparation. Direct examination or direct wet mount of clinical specimens should be performed as soon as possible after collection. Examples of specimens examined by direct wet mount include sputum, drainage from lesions, body fluid aspirates, stool, vaginal discharge, and urine sediment. Examples of pathogens identified by direct wet mount include the motile trovocytes of Giardia lamblia in stool, Trichomonas vaginalis in vaginal discharge, or urine sediment and Entamoeba histolytica from a liver abscess aspirate. So this is going to tie into your stool specimen collection and transport. So this is a broad statement, as soon as possible after collection, but when they get into specifics like CSF or different types of body fluids, you need to know, what are my thresholds, like how long can it stay out, can it be refrigerated, does it need to be at room temperature? These are all things that you're going to need to know for your test. It's extremely important that you know it. Bacteria pure culture, so the choice of nutrients include, oh my goodness guys, I cannot believe it is 2:54. We are not going to be able to get through all of this content. There's too much to know. There's too much to know in life. Okay, the choice of nutrients included in the Petri dish and the incubation conditions are critical to the growth and identification of bacteria. So when you take a micro class, they've got these different incubation sections, okay? And they've got them set to different temperatures. And then make sure that you know the temperature that you're gonna be putting your plate into, because, oh wait, what happened? Did I click on something? Oh my goodness. They're trying to kick me out. Please hold. Oh, there we go. Okay, yes, like I was saying, temperatures, very important. And then the type of nutrient that you choose. Okay. It's being difficult. Okay. Just be nice. Okay, so the choice of media depends on the site that's being cultured, whether it's throat, blood, or urine, the growth requirements of common or suspected pathogens, and then the likelihood of normal flora. So let's talk about our growth media. Nutrient agar. Can somebody give me an example of nutrient agar, or what we use nutrient agar for? Okay. Okay, TSA, yes, general growth. Jarvis, 10 out of 10. Jarvis Strickland. 5 points, 5 points to Jarvis. So it supports the growth of a wide variety of bacteria. An example would be trypticase soy agar with 5% sheep's blood. Okay, next, let's talk about enrichment medium. It contains special nutrients necessary for the growth of fastidious bacteria. Fastidious, what does fastidious mean? Hard to grow, very good. Picky, I like that, yeah, picky, hard to grow, difficult. Okay, Nahid's telling me chocolate agar, very good. Chocolate agar utilized for the growth of Neisseria meningitidis. All right, selective media. Selective media. What are we using selective media for? Okay. It contains chemicals or antibiotics designed to inhibit normal, common commensal bacteria, such as bismuth sulfate agar for the isolation of Salmonella species. Differential media. This is going to stain our colonies of specific organisms while inhibiting the growth of others. Acetate agar to differentiate between E. coli from Shigella. Okay, so important for you to know your growth medias. I don't know if you're going to have to do some study cards or some games or however it's going to help you to remember this. Like I said, there's nothing like spending some time in a lab, or if you don't have a lab, YouTube. All right, YouTube will help you. YouTube has gotten me through so many certifications. Because you think that there aren't people who aren't interested in this kind of stuff. Let me tell you, there are entire channels dedicated to specific portions of the work that we do. Like there's this guy called the Sterile Guy on on YouTube, which he always jokes, you know, he had a vasectomy and like that's why it's his title. But no, honestly, he is all about sterilization. It's literally all about sterile processing. So guarantee you there's tons of channels that will just focus on lab concepts. I know they're out there because I've watched their videos. There's no excuse. The information's out there. Okay, next we're going to talk about the effects of oxygen on the growth of various types of bacteria. They do have multiple questions on the test that talk about obligate anaerobes, facultative anaerobes. They want to make sure that you understand this concept because oxygen is very important, and it applies to even it's something that's as important as our blood culture collection, right? So when a provider orders a set of blood cultures, right, or a blood culture draw, for our adults, it's gonna contain 2 sets of bottles, right? So 4 bottles total. And guess what? You've got to collect 2 bottles per set. Why? Why do we have 2 bottles? One's aerobic, one's anaerobic. Yes, Julia and Eva, great job. Aerobic and anaerobic. So all of this stuff plays a huge role into the care of our patients. I mean, you have to realize that it goes beyond just like testing, right? It goes beyond just like, I need to know this information to pass this test. No, you need to know this information to make sure that you're practicing at the top of your like knowledge as an infection preventionist. You're one of the most knowledgeable people, you know, in the hospital as far as all of the different departments, how they work together, how they collaborate. There's so much that we need to know, right? Okay, so obligate aerobes, effective oxygen on growth, only aerobic growth, meaning oxygen is required. So you can see your little tube, all of your bacteria are at the top and they're like fighting each other, making sure that they can get the oxygen, thrive and survive. Growth occurs only where high high concentrations of oxygen have diffused into the medium. Explanation of oxygen's effect. You know what? We're not going to go into this. We're not going to go into this. We don't have enough time. We're running out of time. So facultative anaerobes. They're both aerobic and anaerobic in growth. There's greater growth in the presence of oxygen, but it occurs throughout the tube. So you can see that distribution. It's all over the tube. And by the time that you're down here and it's anaerobic, they're switching over to fermentation. All right, obligate anaerobes. Only anaerobic growth. It ceases to exist in the presence of oxygen. Again, the importance of why we have those two different bottles that we're using, our aerobic bottle, our anaerobic bottle. Growth occurs only where there is no oxygen. You can see they're all at the bottom of the tube. Aerotolerant anaerobes. Only anaerobic growth, but continues in the presence of oxygen. So growth occurs evenly, oxygen has no effect. And the very last one, which is microaerophiles. Only aerobic growth, oxygen is required in low concentrations. Okay, so let's do a concept check. A microbe that can grow in the absence of oxygen, but is also able to utilize oxygen for growth is A, or an. A, aerobe, B, obligate anaerobe, C, facultative anaerobe, or D, microaerophilic anaerobe. All right, and that is a facultative anaerobe. We have it right here. A microbe that can grow in the absence of oxygen. So we've got our absence of oxygen down at the bottom. Okay, but can also use oxygen to grow. This is an example of a question that they could ask you on the test. All right, so let's do some questions so that we can go ahead and end this. Microorganisms are grown on culture media made of an agar base. Additive to media vary according to growth requirements of organisms and or the desire to select out a specific organism. Fastidious organisms require blank media, and blank media is used to inhibit normal commensals. Getting lots of different answer choices here. I've got some A's, some B's, some C's, a D. Okay, we've got all answer choices. All right, so the correct answer is B. Fastidious organisms require enriched media, and selective media is used to inhibit normal common commensals. And that's when we go back to this here, right? Knowing our nutrient agar enrichment medium, our selective media, or differential media. Okay. All right. Fastidious organisms require enrichment media. Which media is used to inhibit common commensals? This should be an easy one. We still have some people getting it wrong. Okay, it's going to be selective. So, hey, you got to start catching yourself. If I'm missing these questions, guess what that means? I need to go back. I need to read. I need to gain a better understanding of my media and what they're used for. An example of a selective media that inhibits gram-positive bacteria is. An example of a selective media that inhibits gram-positive bacteria is. Okay, so somebody said, I have no idea. I always tell people this, when you're taking an exam and you have no idea what you're looking at, you have to ask yourself, what do I know? What do you know? So there has to be at least one piece of information in your brain that you know. If you don't know anything, take a guess, keep it moving. We don't have time. We don't have time for this. So guess what? Tryptocase soy agar, we know is a nutrient. It's just your regular schmegular TSA. It's what we use in the, in the general microbiology classes to grow stuff. So we're already going to know it's not selective. It's nutrient. It's just to help the girlies thrive, right? It's just helping the girlies thrive. Is it really helping you narrow things down? No, no, no. It's a club with no cover fee, okay? You're just gonna go in there, have a great time. Now, chocolate agar, right? What can we use chocolate agar for? Neisseria meningitidis, fastidious organisms. That's not gonna be selective either. Sabouraud agar, you don't have to know this. I think it was 1882, fungal. If you don't know that, that's fine. At least that lowers it to A and D, and now you have a 50% chance of getting it correct. Correct answer is McConkey's agar. That's going to be our selective medium. Okay. All right, guys. It is 15:06. We did not get through the whole presentation today, and that's okay. We will go ahead and stop here. So next week, I'm going to send out an email because like I said, I'm going to have to rearrange the time because next Friday I have a very big meeting. So I'm going to try to do Thursday. If for some reason I can't do Thursday and we just have to, you know, meet the following week, don't worry, you'll get an email from me over the weekend, and I'll also post on LinkedIn because that's the easiest way for me to communicate with. How do I do this? Oh, you're asking me for the links. Yes, I will. Let me try to see if I can do that now, if I can get my life together here, because I can't figure out how to stop sharing my screen. Like maybe this, stop sharing screen. Oh, there we go. Yeah, well, that makes sense. Okay, so I will go ahead and post the links. I will let you guys know when we can meet, when we can't meet. Like I said, the easiest way to get a hold of me is going to be through LinkedIn. You can just message me on there and I'll respond. I only really check my CIC EPI's email when I have time, which you know, time is difficult. Okay, so there's the Google Drive link, and I will also post the links do not appear in the recorded session. Is it possible to include them in the video description? Yeah, I can definitely do that. So I will, oh, let me stop recording. Stop your recording.